And I'm looking forward to talking about mycorrhizas and prairie restoration with everyone. Um, so when we're thinking about plants, it's important to think of their interactions with soils, right? And I think that that includes the physical component, which many people are aware of, but also really the living components of soil as well, including diverse microbes. So we have plant growth promoting both bacteria and fungi, also the endophytes that are found within plants, the mycorrhizal fungi, which is the group that I often focus on. And then, of course, there are also negative interactions, such as with pathogens. Thinking about arbuscular mycorrhizal fungi, most plant species are going to form associations with these fungi. And so what you're seeing on the left is a plant and its root system, and what you can't see is shown on the right, which is what those AM fungi look like as they're growing as hyphae in the soil, and then also colonizing plant roots, forming vesicles and arbuscules, which are exchange structures between the plants and the fungi, and then, of course, the very fine hyphae that are colonizing the soil. When we think about the roles of AM fungi in terms of their benefits to plants, the primary function is the way in which they enhance plant nutrient uptake, particularly of phosphorus, zinc, copper, and to some extent ammonium. They also have been shown to improve plant drought tolerance. They seem to play a role in protecting plants from pathogens. But a really crucial thing that gets forgotten or overlooked sometimes, I think, in thinking about benefits is that there's also a cost to the plant. Arbuscular mycorrhizal fungi are not able to synthesize their own lipids, and they aren't able to acquire energy from their environment in the way that other fungi do. And so they're dependent on the plants for that energy that comes from photosynthesis. And I just wanted to include here a plant that's of special concern in Minnesota. This is Gaillardia aristata, or blanket flower. And so what we see is, and if you look at the graph on the left, what we see here is that a typical pattern that we see for plants, and this is especially demonstrated by blanket flower, is that the more AM fungal hyphal length you have in the soil, so the more colonization you have of the soil, then the greater you see the phosphorus content in the plants. And then if you look at the graph on the right, you can see that that increased plant phosphorus translates into increased biomass. So a very simple relationship between the formation of mycorrhizas between the plant and the fungi, and then a benefit to the plant in terms of phosphorus uptake and ultimately biomass growth. When we think about the ways in which AM fungi benefit plants, there's a lot of complexity there. So here I'm showing you work from Koziol and Beaver, where they're on the x-axis, you can see they've divided what they're calling sort of early successional plants and late successional plants on the right-hand side of that graph. And then the colors represent different species of AM fungi. And so they're looking, and these are actually at the level of genus for the most part. And then this is a log scale of mycorrhizal responsiveness. And what that means essentially is how much additional biomass the plants are acquiring as a result of the formation of the mycorrhizal association. And so you can see this general pattern where for early successional plants, there doesn't really seem to be a clear benefit to forming these associations. But on the right hand, you can see for these later successional plants that in general, there is a clear benefit in terms of biomass, and that benefit varies from AM fungal species to AM fungal species. So they're not all the same. And then hopefully this is something that all of you are familiar with already, is this general problem that we have in prairie restorations where only a fraction of the species that we seed actually establish. And so these are data from Diane Larson and her colleagues looking at Glacial Ridge National Wildlife Refuge 4 to 14 years after they seeded different prairie reconstructions. And so you see the planted species richness on the x-axis, and then the proportion of those species that they actually observed established either 4 to up to 14 years later. And so you can see that at relatively low richness of seeds, so relatively few different types of seeds, you get a reasonable proportion of those species then establishing. But as you increase that richness, the picture gets worse and worse. And so it really is a challenge to figure out how do we get prairie restorations that are as diverse as remnant prairies. And you can see that in this map on the right-hand side, where you can see the pre-settlement extent of tallgrass prairie, and most of you know that there's only a tiny fraction of that left. So that brings me to kind of the major questions in my research, just thinking about to what extent are AM fungal communities actually different between remnant prairies and prairie reconstruction sites. In other words, do we actually see differences, and then are those important to mitigate? And so if there are differences, then do they actually influence prairie plant establishment? Do they play a role? And finally, then, should we actually be inoculating plants with AM fungi as part of prairie reconstruction? So tackling this first question, do AM fungal communities differ between remnant prairies and reconstruction sites? There's been quite a bit of work on this, including some work that I did with the people shown here in these photographs. And then you can see on the right-hand side these different paired sites where we essentially have these prairie reconstructions. They were all established by Diane Larson's group in 2005 using the same seeding techniques and management. And then we were able to locate remnant prairies that were nearby, and we sampled in 2020, so essentially 15 years after the prairies had been reconstructed. And so we have these 6 pairs of sites. Sometimes we have two reconstructions and then a single remnant prairie in one of these groups. And when so these are samples of AM fungal communities, and essentially the closer together 2 points are on those graphs, then the more similar those communities are. And so on the left-hand side, I'm showing you the origin of the soil that these plants were planted in. And if it's in green, these are remnant prairies. And if it's in orange, then these are reconstructions. And so you can see, hopefully, that the plants that are inoculated with prairie soil from remnant prairies show much more diverse communities and distinct communities from the plants that were inoculated with reconstruction soil. So we can say that these AM fungal communities are different. But there's an additional nuance to this, which is that the communities also differ by the plant species. So if you look at the graph on the right-hand side, what you can see is in orange, I'm showing you different grass species, and then in purple, different forbs, and then Dahlia purpurea is our only legume. I'm sort of signaling out in green. And so again, what we see is that there seems to be this general pattern where forbs are associating with these diverse communities, and then grasses a somewhat less diverse community because those points are clustering more closely together, and that community is generally distinct from the community in forbs. One thing that surprised us in this study was that what would we see in terms of just the number of the different types of AM fungi that are found in reconstructions and remnants? And so this is a very bad Venn diagram because it's not scaled correctly, and I apologize for that. But what I want you to notice is that we actually documented 120 sort of species of AM fungi at these sites. So this is a very diverse community. 72 of those species were actually shared between reconstructions and remnants. So even though the communities are distinct, there is a lot of sharing of species. But also you'll notice, right, that there are actually fewer distinct species for the remnants. In other words, species that we only found in remnants. We were more likely to find species that were unique to reconstructions. So there's this shift in the AM fungal community, and there's actually an increased richness of different types of AM fungi in the reconstructions. But are those reconstruction AM fungi as beneficial? Right? And one thing that we notice when we look at individual plants in reconstructions versus remnants is that there's a small difference in how many AM fungal species individual plants are supporting. So when you look at plants in remnants, on average, they're supporting about 3 AM fungal species. When you look in reconstructions, it's more like almost 4.5. So does that then suggest, to some extent, maybe a loss of specificity, if there is any specificity in this relationship? And also, what does that mean in terms of cost? Is it more expensive for plants to maintain more AM fungi? So to answer this first question, I think that we've shown, and other studies as well have shown, that there is this difference in AM fungal communities between remnant prairies and reconstruction sites. And that actually the remnants are less diverse, and I think there's a tendency to think that that is somehow bad. But I think it's important to consider that that might reflect an increased specificity of native prairie plants to their AM fungal associates. And that specificity may actually be important. And then again, it's important to remember that the relationships are not the same across plant groups, and there are these really distinct differences between grasses and forbs. So moving on to my next question, do these differences that we see actually influence prairie plant establishment? So I'm not the only researcher working on this, but what I thought I'd show you is just briefly a recent experiment by one of my master's students, Erin Semington. And so what you see here is a prairie reconstruction, not really, but a field that NDSU begrudgingly let biological sciences use. And so this has been an experimental field at NDSU for the last 50 years. And what Aaron has here are these mini greenhouses, and he wanted to look at the effect of drought, which I'm not going to talk about today. But what I want you to notice is that for each of these little mini greenhouses, you can see a white plot at one end and a white plot at the other. And so those are paired plots, and one plot received a thin layer of that same field soil that you see there on top. And the other plot received an equally thin layer, but of soil from a remnant prairie. And so we did that in each of 10 greenhouses and then monitored establishment of prairie plant species. And so he used a seed mix that was commercially available from Prairie Moon, and I'm just listing that here and not expecting to look at all this information. But the main point is that in this first growing season of the 18 species that he seeded in, half of them emerged. And he seeded at 4 times the recommended rate because he needed this to work, the first time through as a master's student. So what did he find? And so the first thing that he looked at was emergence, and he looked at both natives and he looked at weeds. And so I'm showing you the data from 2 weeks after sowing, and field on the left, prairie on the right, and the weeds are in purple and the native plants are in orange. And so if you look at the weeds, you can see that there's no difference in weed emergence between the 2 types of soil, but the natives are emerging at a higher rate in the plots to which we added prairie soil. So that actually doubled emergence at 2 weeks, and that pattern held. And then at the end of the growing season, when he harvested, he looked at the biomass species by species. Those of you who've done this know this is a ton of work. So I'm showing you above ground dry mass, essentially a measure of performance for these plants. And these 2 different field inoculum types, where they're either inoculated with the field soil from the reconstruction site, or they're inoculated with the prairie soil. And then the colors reflect the different plant species. The blues are the native plants, and then the reds and oranges are the weeds, the undesirables. So basically what you can see is that there was greater biomass overall in those plots that received a thin layer of prairie soil. And that prairie soil benefited 2 native species, Rudbeckia hirta and Helianthus maximiliani, in terms of their biomass. So it had this positive effect on 2 of the native plant species, and then it had a negative effect on 2 of the dominant weeds, these 2 different foxtail species. You can see that reduction in the size of the red and orange bars. And then interestingly, yellow sweet clover only showed up in the prairie inoculated plots. And that may have to do with the fact that the rhizobia with which they associate were present in that prairie soil. So one quick thing before I move on from this is just that we were measuring soil water content as well as some other variables over time. And so I just want you to look at the 3rd graph down here, the soil water content. And what you can see is the orange line is the plots that were that contained the field soil only, and then the green line is the plots to which we added prairie soil. And you can see that after rain events, of course, they had comparable moisture, but then over time, as that water was drawn down, it was drawn down more rapidly in the in the field soil than in the prairie soil. So the prairie soils maintained higher water content for longer. And that might be a direct effect of the inoculum in terms of the hyphae growing in the soil, which slow water loss and maintain water moisture in those soil surface close to the soil surface. But it also could be an indirect effect of how different these plots were to each other, especially by the end of the growing season. So on the left here, you can see a prairie plot, typical prairie plot, and on the right, field soil. So prairie plots with lots of forbs, quite a bit of shading of the ground, slowing evaporation in contrast to the weedy plots where the sun could reach the ground more easily. So thinking about this question of whether there are differences that might influence prairie plant establishment, we certainly saw that, but this is a whole soil effect. So it's not necessarily due simply to AM fungi. And so we saw this improved soil water content, improved native emergence, and the suppression of weedy grasses when we added this very small amount of prairie soil inoculum. So that brings me to the last thing before I wrap up, which is whether we should not be inoculating plants with AM fungi in restorations. And so here I want to just briefly touch on some data from Aaron and then move to the last little series of graphs. So this is in a growth chamber, not in the field, and looking at purple coneflower. And here we were looking at the effects of temperature. So you can see that on the x-axis here, ambient temperature, which is sort of historical averages, and then using growth chambers to raise that temperature over the growing season steadily by 4 to 6 degrees or 8 to 10 degrees. And then we also have the different types of inoculum. So basically an autoclave blend where there's very little colonization by AM fungi. And then the orange is field soil, the same field soil I just showed you for the previous experiment, and then the prairie soil. So again, we're using a tiny bit of inoculum and all the plants are being grown in this same autoclaved blend. The inoculum is simply to allow colonization by AM fungi. And so we see this pattern where in the field soil, the purple coneflower shows this consistent and fairly high colonization by AM fungi into the root system. And it doesn't change with temperature. But in the prairie soil, you can see this steady increase with temperature. So as temperature increases and as purple coneflower responds positively to this temperature by growing more, you then see this concomitant increase in root colonization, showing this response or this interaction between the AM fungi and the plants that is absent when they're inoculated with that field soil instead. So showing a difference in how that interaction works in prairie soil and in field. So the last little thing I just want to touch on is thinking about if you do inoculate, how does the timing of that inoculation influence plants? And so this is work done by a different grad student in my lab, Ankita Sawant. And so she wanted to know, does it matter whether you germinate plants with or without an inoculum for AM fungi? And if you delay, what happens if you delay inoculation until the seedlings are actually established? Does that change plant performance? And then finally, do we see sort of a consistent response, or does it depend on plant species? And so what she did, and so I'll just walk you through this, is she had she germinated seeds in soil without AM fungi and then soil with AM fungi. And so she had these two groups of seeds that had then germinated and grown into seedlings. And then those seedlings were either transplanted at 11 weeks into pots that did not contain any living inoculum. They just had autoclave soil, or pots in which there was living inoculum. And so that left her with 4 treatments, right? So they either were mycorrhizal and then went into mycorrhizal soil, or they were non-mycorrhizal and then went into mycorrhizal soil. And then, of course, they could start out mycorrhizal, go into non-mycorrhizal soil, and then finally start out mycorrhizal and go into mycorrhizal soil. So those are the 4 possibilities. And so she looked at 3 different plant species. And so this is the pattern for Echinacea purpurea, for purple coneflower. And so what I want you to notice is that she's showing you both above and below ground biomass, so you can get a feel for the total plant performance. And you can see that when the plants are colonized by mycorrhizal fungi, they grow much larger. But that if you actually delay colonization, the plants grow even larger, right? And those plants in particular are investing more in their roots. So there's this advantage to actually delaying the colonization. Now, when she looked at smooth blue aster, she again saw a benefit to formation of mycorrhizas, but it didn't matter whether they were inoculated early or they were inoculated late. And then for Cydodes grama, she saw that same pattern again. Colonized seedlings are investing more in their roots, but that colonization is beneficial whether it occurs early or late. So getting back to this question of whether or not to inoculate plants with AM fungi, I think we have to tread really carefully because we don't know enough yet. That's the bottom line. AM fungi are definitely present in field soil. It's not that they're absent from reconstructions, but they may be more likely to act parasitically. And we don't yet know how to manipulate that relationship. We also know that different plant species associate with different AM fungal communities. So we really need to think about how we would develop an inoculum that would be appropriate across multiple species, or maybe for particularly targeted species of interest. And I think if we're thinking about seedlings in nursery settings, that initial growth performance that's higher without AMF, it reveals and reminds us of the cost of AMF. So again, the timing of inoculation may depend on the species that we're working with. And all of this points to this urgency of developing a true sort of prairie inoculum, not maybe just AMF, but also including the other organisms that are clearly beneficial for native plant establishment. So to conclude, I'd just like to say that AM fungal communities do clearly differ between reconstructions and remnants. Reconstruction AMF may be less beneficial to at least some native plant species. I want to remind everybody that current inocula that are commercially available can be deeply problematic. Those are often very species poor mixes, and they also are not necessarily species that confer benefits to plants. So I think we really need more field experiments with practitioners. I'm really interested in cover crop herbicide treatments that may allow us to shift reconstruction AM fungal communities in a way that would make them more beneficial to native plants. We have this question of whether you can use plugs of nursery grown inoculated seedlings to sort of form loci in the field that may facilitate establishment of beneficial mycorrhizas. And then an ongoing question for me is the extent to which we can use inocula to actually reestablish those rare plants that are so difficult to establish right now in prairie restorations. Thank you very much. And I'll turn it over to the next speaker.
Thank you, Laura. Sai, do you mind sharing your screen? Thank you, Sai. So thank you all for giving us the opportunity to talk about our project, Comparison of Organic and Proprietary Soil Amendments for Vegetation Establishment. I'm Bora Cetin from Michigan State University, and Sai and I will co-present next this presentation. And Sai was one of the PhD students work on this project. We have a big team. We collaborated with University of Maryland on that, and the funding came from Minnesota Department of Transportation. And I will acknowledge our entire team at the end of the presentation, and of course all the technical advisory panel members from Minnesota Department of Transportation. Next slide, please. Slide. So the specific objective of this work is on the roadside vegetation establishment. The vegetated roadsides in Minnesota occupy approximately more than 24,000 acres of land. And what happens most of the time with these roadside constructions happen after construction is completed. These roadsides are generally unvegetated and being exposed to rainfall and subjected to the soil erosion. And in addition to that, with the current increased numbers of extreme weather events, especially stormwater events happening, once you build, the construction is completed, and if you don't establish a good vegetation establishment as quickly as possible and as long lasting as it can be, it causes significant soil erosion problems and stormwater pollutions. Next slide. And again, just to reiterate some of the things that I said, if you don't have good, quick vegetation establishment on these roadsides, that soil erosion on exposed land will lead to the sedimentation in water bodies. This disrupts aquatic ecosystems and causes eutrophication due to sediment and associated pollutants. Even though majority of the work was not specifically focused on the pollutants, as part of the work, we still did some of these, look into some of the pollutants. Next slide. Yeah, next slide. So one thing I also want to say, introducing vegetation along these areas can turn the tide on this, and plants act as natural filters. They improve runoff water quality, reduce runoff volume, stabilize slopes, and can improve carbon sequestration. And all of it together makes vegetation a cornerstone of preventing soil erosion. And this is not just beneficial for landscape aesthetics, but it's also a vital step in landscape management. Another thing, what happens in the post-constructions, there have been applications there that on the top you either put very good top quality soil, which is not commonly available, or especially when it comes to the roadside construction. They put fertilizers, straws, and turf grass, but fertilizer has some environmental issues depending on the area, and turf grass and all that can be very expensive solutions as well. So in this project we wanted to come up with methodologies, especially using either compost or proprietary amendments to see if we can improve this vegetation growth and vegetation establishment, and come out with these optimum mixing ratios to determine a cost-effective solutions, as well as that provide good vegetation establishment and has minimum environmental impact. Next slide. So what are the two, again, just to reiterate, the two things that we wanted to test on this project, which is almost about to be done in a couple of months. We're wrapping up our final report to tasks. We wanted to test different type of compost. We tested 3 different composts, which we call as organic amendments, and biochar material as an amendment to the organic compost additions. And the other material that we wanted to test is the proprietary amendments. These can be costly due to location, but we wanted to see if some of these proprietary amendments can be used, and depending on the location of the day, may be an alternative material to use the roads to improve roadside vegetation. And overall issues with these were there are no set guidelines. Even though there are some sort of guidelines, there are no specific guidelines. So we tried to come out with these maybe specifying some already existing overall guidelines to use these compost, biochar, and proprietary amendments. Next slide. And again, the overall objective is, like I reiterate some of the things that I said, is to determine the optimum mixing ratios for both compost and proprietary products used as a soil amendments to rapidly establish this post-construction vegetation on the roadsides. And the 2 main research objectives under this overall goal is to evaluate the use of organic. Again, organic part includes compost and biochar, and proprietary amendments for this roadside soil remediation without having significant environmental impacts. And we have conducted greenhouse pod studies, which we have tested approximately 300 pod studies on 150 to 150 on each institution, with different rate mixes of compost and proprietary amendments with 4 different type of soils. And then on we select based on the pod studies in greenhouse, we selected the most promising and worst case scenario mixes, run greenhouse mesocosm studies, and look into the impact of the vegetation establishment and erosion soil erosion under heavy rain events. And then from the mesocosm studies, we built test sites, test plus 12 of them on one site to check to see how it would perform in the real field conditions in Minnesota. And then the second objective was digest all this data that we collected from them, first objective, and do some analysis and compare the benefits of using organic and proprietary amendments for vegetation establishment on the roadsides. Next slide. And due to the limited time that we have, and we talk about this with Wade and Carly as well, we'll be able to give another webinar talk. Like I previously said in the objective slide, we have pod studies, mesocosm studies, field study, lot of different rates with different type of soils, mixing their performances. But in this study, we will specifically focus on the organic amendments, particularly on the compost mixes with some of the soils that we tested. So we won't get into much details on the pod studies in greenhouse and proprietary amendments, and not much information about the field study data on this webinar. But again, we'll be happy to give webinar talks on the other the parts that we're omitting on this current webinar. Go ahead, Sai. And after this slide, I'll turn that over to Sai. So benefits of soil organic matter was one of the main parameters we look into the selection of compost and biochar. The three type of compost that we use: food waste, yard waste, and turkey litter compost. And one of the main requirement of the MnDOT roadside soils were organic matter content. It needs to range between whatever the mix or soil that you have, the organic matter content has to be between 3 and 15%. It didn't have any criteria for nutrients. And from here, I'll leave it to Sai. Sai, continue, please.
Thank you, Dr. Cetin. So as he mentioned, I worked on the organic amendment side of things for this project. So I will be mostly talking on that today. One of the indicators of soil health is the organic matter content. It is important that the soil has an adequate amount of it in order to support vegetation growth. So how can we improve soil organic matter? Now that will be through the addition of organic amendments. These are basically materials developed from plant or animal matter, or even stuff like sewage sludge and food waste, for example. And depending on what process they undergo, they can end up becoming amendments like compost, biosolids, biochar, mulch, all of which can enhance soil organic matter and structure. So before diving into the mesocosm experiment, I'll quickly present the highlights from one of the first experiments we did, which is the pot study, as it was crucial in our decision-making process. So in our pot study experiment, we tested 3 types of composts, Dr. Cetin already mentioned, that were derived from yard waste, food waste, and turkey litter. And we also evaluated one wood-derived biochar as the 4th amendment for vegetation growth. Now, these amendments were applied at 3 different rates to finally increase the organic matter content of the control soil to 5, 7.5, and 10%. And all of these numbers are within the range that's specified by MnDOT. The soils used in the study were also sourced from Minnesota. Now, this study was undertaken to determine the optimal mixing ratios of soil and organic amendments to improve vegetation growth. Overall, as you can see, yard waste amendment positively influenced the growth metrics like plant coverage, biomass, leaf area, while biochar had a clear negative influence. Now, this is due to the low nutrient levels that biochar possesses, particularly nitrogen. For the other 2 composts, the turkey litter compost also stunted growth severely, and especially at higher rates of application, as you can see in the picture on the right here. This is because it contained high salts and ammonia contents and it caused plant toxicity. Food waste produced growth, but then it was at a slower pace. So overall, the yard waste amendment was chosen as the best among the 4 that we used in the pot study for vegetation growth. Now moving on to the main focus of the presentation, the greenhouse mesocosm study. Based on the pot study and literature, we know that the yard waste compost performs well in terms of vegetation. But there are concerns raised with the use of this with regards to nutrient water quality. On the other side, there is well-documented science that supports biochar as an excellent amendment for improving water quality. So the idea of this particular experiment is to put the 2 amendments together in a pilot scale study, which simulated field conditions in order to identify their synergies. So the mesocosm experiment ran for 3 months. We simulated 12 weekly rainfall events of 2.54 centimeters depth or 1 inch on soil mesocosms and measured various water quality parameters from the collected effluents. We also monitored vegetation coverage every week, and other measurements like biomass, tissue analysis were taken at the end of the study. For the sake of time, I will discuss only the start parameters here, which focus on nutrients in water and plants. So we tested 4 amended soils against a control soil that is represented as C. The first amended soil is mixed with biochar alone, which is CB. The second one was mixed with only compost, that is CY. The third was with equal parts of compost and biochar, that is represented as CYB. And the fourth amended soil is with 3 parts of compost and 1 part of biochar, that is C3YB. Now these amendments were mixed to increase the organic matter content of the soil to 6.5%. This plot shows nitrogen leaching over time. X-axis shows weekly simulated rainfall depth, and y-axis is the amount of nitrogen that was transported into the water effluent. The plot suggests that with increase in the biochar's contribution to soil organic matter, less nitrogen leached into the effluent. Whereas on the other hand, compost has incurred more nitrogen losses with the mixtures CYB and C3YB lying in between. Now, why does this happen? One of the most important parameters in the context of nitrogen cycling in soils is the carbon to nitrogen ratio. This is predominantly discussed in terms of nitrogen uptake by plants, but it can also be applied to nitrogen leaching. Biochar drives up the soil carbon to nitrogen ratio as it is a carbonaceous material, and in such a scenario, the soil microbes tend to immobilize the nutrient in order to maintain their nutritional balance, because there is more carbon compared to that of the nitrogen in the soil. And this is why we noticed low nitrogen leaching from the biochar amended soils. On the contrary, when you have more nitrogen in the amendment, in this case it is the yard waste compost, you drive down the CN ratio and it favors nitrogen mineralization, contributing to greater export of the nutrients from these soils. The story kind of remained consistent even with the phosphorus nutrient. Amending the soil with biochar had a positive effect in that less phosphorus leached out of the soil, whereas the yard waste compost amendment contributed to a higher release of phosphorus. However, in the phosphorus discussion, it is important to note that both biochar and compost increase the soil phosphorus content, yet biochar contributed to lower P losses due to the retention of the nutrient in the pore spaces of biochar and also by the soil minerals. Also, the amount of biochar we add in order to increase the soil organic matter at the same rate is lower compared to that of compost. This overall made biochar more favorable in terms of water quality. So while biochar was our clear hero in terms of water quality, it was not so much in the case of vegetation. You can clearly see that the vegetation was stunted in the CB soil. This is showing the coverage here. And the other 3 soils had better coverage because of the yard waste amendment. So CYB, C3YB, and CY. So regarding the effects of these amendments on plant nutrient uptake, firstly on the top, you can see that the addition of compost has actually increased the plant nitrogen content, and biochar reduced it, similar to that of the coverage observations. On the contrary, with the phosphorus, we found that among the compost amended soils, that is CYB, C3YB, and CY, the greater the addition of biochar, the greater was the phosphorus uptake. This shows that biochar not only reduced phosphorus in the effluent, but also improved its availability for the plants to utilize this nutrient better. So between the vegetation and the water quality data, this growth study underscored the positive influences of combining compost and biochar to improve soil organic matter and thereby restoring the roadside soils for vegetation growth. So in conclusion, when we have the yard waste compost amended into the soil, it supports soil health and vegetation, but it comes at the cost of higher nitrogen and phosphorus losses to surface or groundwaters. On the contrary, when you add biochar, it increases the nutrient retention in soils, but it does not favor vegetation growth. Now, when you have compost and biochar mixtures at the rate that we applied, you can achieve this equilibrium state where plants get the nitrogen and phosphorus they need from the soil without compromising water quality. I'll hand it over to Dr. Cetin right now.
Thank you, Sai. Next few slides, the translating research, just like we put there from greenhouse to ground, we built 9 plots, with each of them 3 replicates. So 3 different, 1 control soil, and 1 with one of the selected proprietary amendments, and 1 with the selected compost rate and compost type. And we have monitored the sites for almost 2 years at the Minrod facility in Monticello, Minnesota. And again, we won't get into much of these. We won't be able to get to the results of this, but we look into the total nitrogen, the total phosphorus, and pH, electrical conductivity measurements, and did all the vegetation growth, monitoring vegetation coverage, biomass growth, and all that. And hopefully in another webinar, if we get a chance, we'll be happy to talk more detailed on the field applications. Next slide, please. Sorry. And great, we really appreciate Minnesota Department of Transportation for funding and supporting this study. We're wrapping up the final report and final report task. So hopefully by early spring, the report will be available for the public. And we pretty much, we really thankful to all the technical advisor panel members from the Minnesota DOT. Special thanks to Warren Twill, who was the technical liaison, and Dave Glyer, who was the project coordinator, and Jeff Tabry from Minrod really helped us to collect data and send the ship the data to us for running all these tests and helping us to monitor. And of course, all from Ken, Dave, Jim, Tom, Tim, Chris, and Dwayne and Annie to help all their input and guidance throughout this project. Next slide. And of course, we had a big team. I appreciate my colleagues, other co-PIs, Dr. Eilek and Dr. Davis from University of Maryland, and Dr. Choban, Dr. Farina, the postdocs who involved in this project. Still, Dr. Farina is with us. Dr. Choban moved to Wisconsin DOT. We really appreciate their help on this work. And of course, the most thanks to Dr. Pamaru now and our current PhD student, Oğuzhan Saltalı. So next slide. Sorry. And that's pretty much it. Thank you again for giving us the opportunity to present at least some portion of our work.
Thank you all for your presentations. Really appreciate that. We do have a bunch of questions, so we're going to dig right into those. I'm going to throw one to Laura first. Just a question about the do commercial inoculants vary much for the mycorrhizal species that would be used or that would be found and tend to dominate in certain groups? And why are some of those commercial inoculants so harmful or rather unproductive for restoration and germination of seedlings?
Yeah, great question. So a lot of the commercial inoculant is a single species, and it's gone by the name Glomus interradices. It has a new name now, but that's the main species. It's extremely easy to grow and get it to sporulate. And so the concern is that it's actually acting as a parasite. It's colonizing the plants, essentially at their expense. And we see that when we test it for growth benefit to prairie plants, we've yet to observe any.
Thank you. That's very interesting. I never thought about that parasitic aspect of it. Have you looked at the diversity of AMF in reconstructed prairies over time? And does the diversity decrease as the reconstructed prairie is managed maybe with regular disturbance regime, for example, fire? Do you have any sense of how those interact?
So I haven't worked on that personally, but there is some nice work in the literature using chronal sequences, and in general, we see increases in diversity with time and with disturbance, and I think that's because the AM fungal diversity is tied quite closely to the plant diversity. So of course, if if it's a prairie with a fire regime, then maintaining that fire regime will be important for that diversity. So I think it really depends on the types of disturbance. So grazing and soil disturbance by rodents and things like that. That's part of a prairie ecosystem. What's not is tillage, right? So tillage definitely favors AM fungi, again, that appear to be less beneficial to prairie plants. And some of the mechanisms of that are not well understood. That's still a big black box, but we definitely see that pattern.
I'm going to throw another one to you and keep on the theme here. Is there an easy way to assay the fungal communities previous to doing a prairie seeding? If you're trying to do, as part of a site assessment for a project, can you kind of make some kind of assessment of the populations and the diversity?
Yeah, it used to be incredibly difficult, but with high throughput DNA sequencing, we can do it quite quickly. And the price has really come down. My lab does it routinely. So if somebody's interested in how you might go about it and what your budget needs to look like, please contact me. I'd be happy to help.
I think a lot of people will be interested in how much that is for their budget.
Yeah, it's really not bad.
Good. I'm gonna just change it up here. We'll send one over to Bora and Sai. What type of biochar was used for the experiments? And as part of that, can you actually, just in case some people aren't familiar, can you give a brief definition of what biochar is and how you would acquire biochar? You both need to unmute.
You want to start?
Sure. So the biochar that we used in the study is derived from wood. So it's a wood-based biochar. And please let me know if it's wrong, Dr. Chetin. I think this was sourced from the American Biochar Company.
Yep.
Also, it's called the Naked Char, and it's basically like activated carbon, but then it is definitely cheaper than that. It's a carbon-rich material produced during this process called pyrolysis, and you can kind of recycle any of the organic wastes and make it into biochar. And depending on what waste you use, that is going to affect the quality of biochar and then therefore the vegetation growth. But in our case, we used wood-derived biochar, so it had low nutrients in it inherently.
So you could burn anything to make the biochar. It doesn't have to be a tree.
Doesn't have to be just the tree. No, people also use some kind of agricultural residues or manures and so on and so forth.
Okay, thanks. So one of the participants is curious what plants you were using for your seed mixes. Were they very diverse? Were they grasses and forbs?
Yep, yeah. We used one of the MnDOT native seed mixes, which contained both grasses and forbs. Around 7 different species were in it, but predominantly we were able to see the black-eyed Susans emerge out of it compared to grasses. Yeah.
I have the seed mix, specific seed names that we used as the mix right in front of me. I saw the question. So big blue stem native Indian grass, slender wheatgrass, comes from black-eyed Susan native, purple prairie clover, and Canada milk vetch. Those are our seed mixes included in terms of plant seeds. Another
one of the questions that comes in, I'll leave it with you guys. do the biochar compost mixtures, when you use them together, do you have to allow some kind of time for them to, the word was equilibrate or sorb with each other before using them as a growth medium? So do they need some time to kind of acclimate?
I believe so. Sometimes there are tons of studies which are actually looking at co-composting, compost and biochar together, which is kind of that's the scope of their studies. But then in our case, we wanted to keep it kind of simple in that the goal of our study is to improve the organic matter content of the soil. So the way it's done in the field is like you just add compost or whatever organic materials you have, you mix it into the soil and measure the organic matter content. And that's exactly what we did, and then eventually seeded and then monitored growth and water quality through time. So I'm assuming probably within the first 2 to 4 weeks, the soil conditions would have equilibrated. And then that's exactly why in some of our plots, we saw that plateau with the way nitrogen started leaching or the phosphorus started leaching. In short, we didn't equilibrate before seeding, but then the process was more dynamic in our experiment.
And one reason why we didn't wait it with these road construction this is funded by Minnesota Department of Transportation. They really construction crew just laid out and they don't want to wait for, or they don't have time most of the time to wait for that type of equilibrium to be established. So they want to see would it work with their standard construction style or procedure. That's why we didn't dig into that equilibrium time.
Thank you. Is it economically efficacious to apply biochar on high pH soils that are coming out of a continuous cropping system in preparation for prairie seeding? That might be beyond your scope, but let me know if you can respond to that.
I would leave that to Sai because our research main focus, we did some cost benefit analysis, but not that way. I'm not from that perspective. Go ahead, Sai. I'll trust your judgment on this more than mine.
Yeah, biochar, I think it is definitely more expensive compared to compost, especially yard waste compost. And one of the nice things about using biochar on the field is that you don't, especially if you're trying to target to improve the organic matter content, you don't need a lot of it. As opposed to compost, you need half of what compost would do in order to improve that organic matter content. So as just a little bit will still give you that structural benefit to the soil, just like composts. So keeping that in mind, I think it is definitely important to do some level of cost benefit analysis and then, see how the application of biochar turns out in the field. But if the soil is already at a high pH, now that could be limiting in terms of certain nutrients like phosphorus because they tend to complex and they can't really the plants can't really absorb that nutrient from the soils. In that case, biochar might not be the right amendment. So first assessing your soil type and then understanding the base properties of your soils is more important before we choose the amendment itself.
Thank you. Laura, I'm going to combine a couple of questions here, and we're winding down pretty quickly here, but we'll see where you can get with these. But the question would be like, how much, if you were to inoculate using soil from a remnant or something like that, how much soil would you need? And how would you, you talked about, little loci of inoculum versus like broadcasting the inoculum. Like, can you talk kind of about like how much and methods to do that?
I mean, we're definitely not there yet. What we used on our plots it was less than an inch thick because we were really trying to minimize how much inoculum we used. But obviously going to remnants to do that on a large scale is not viable. So I think that's kind of our next big question, is can we develop in the lab essentially something that's at least almost as good as prairie soil inoculum? And then, then we turn it over to commercial interests in terms of trying to actually maintain that. But I think that's the next big challenge. And I've already mentioned this, but Liz Koziol actually has a business. She's based in Kansas, and so she's focusing on tallgrass prairies there. But she's definitely trying to do this and already preparing inoculum. But she's, you know, she worries about people using that elsewhere because then we're essentially introducing new species. And I know most of us are a little bit leery about invasions, right? And we should worry about invasions below ground as much as we do above ground.
Great. We've gotten to 1 o'clock, and so I just want to thank all of our speakers and our audience for your great questions. So great presentations, great questions today. And if you have further discussions or questions for some of our speakers, I'm sure they'd be happy to field those offline. Thanks,
Thank you.
everybody.
Bye,
Thank
everybody.
you.